Ever get kind of blown away by like just how clever life on Earth is? The way things change and shift,
Mm.
adapt to crazy situations, and just explode into so many different forms.
Yeah, for sure.
Today, we're going deep on that ecological genetics. It's this whole field that tries to answer a pretty big question. How do genes and the environment team up to create all the incredible variety we see in the living world around us?
It really is about understanding the why behind the diversity, right?
Absolutely. It's like we see the what all the time, but ecological genetics is all about that deeper explanation. And to help us navigate all this, we're diving into some excerpts from the Stanford Encyclopedia of Philosophy.
Oh, nice.
Yeah, their entry on ecological genetics. So we're starting with some seriously solid expert level stuff. And our goal today, I think it would be awesome to get a grasp on like the main ideas in this field, especially, the back and forth debates that happened historically and also how we understand things now, like how genetic variation sticks around and how adaptation happens out in the wild. I kind of want to get past that simple survival of the fittest thing. Like,
Yeah.
let's really dig into the real forces that drive evolution. Okay, so the entry mentions this thing called the modern synthesis. What exactly was that?
Well, the modern synthesis, it really kind of came together in the early to mid 1900s, and it was a big deal in biology, a huge turning point.
I see.
It was basically the moment when Darwin's idea of natural selection, which was groundbreaking, finally clicked with the newer understanding of how genes work, like Mendelian inheritance and all that. You can think of it like, okay, now we finally get both the engine and the blueprint behind how evolution happens.
Hmm.
The folks who really nailed the math of it were R.A. Fisher, Sewell Wright, J.B.S. Haldane. These guys developed models showing how the frequencies of genes change in populations over time.
Makes sense.
And then there were these other thinkers, Julian Huxley, Theodosius Dobzhansky, and Hermann Muller, who brought in some key insights and really helped spread these ideas to the rest of the scientific world.
Okay, so before this big synthesis, was everybody thinking about evolution in different ways? Like, what gap was ecological genetics trying to bridge?
Totally. You had these evolutionary geneticists who were super focused on the numbers, the math of how gene frequencies shift around
Yeah.
due to things like mutation, genetic drift, selection, and gene flow. Their whole view was like zoomed in on the gene itself.
Interesting.
But then you had all these naturalists and ecologists. These guys were mostly interested in how the environment shapes adaptation, how creatures become so perfectly suited to their little niche in the world. So ecological genetics kind of popped up to be the bridge between these two worlds.
I see.
It was all about figuring out how those changes in gene frequencies actually play out in the real world, like how they turn into adaptations to all the different pressures out in nature.
Okay, that's cool. And so the entry talks about two main patterns in nature that ecological genetics tries to figure out. What are those, like the big areas of focus?
Well, first, there's adaptation itself. How do organisms become so incredibly well suited to where they live and what they do? And then there's polymorphism, which is super fascinating. Is when you have multiple forms of something, either how they look or their genetics, all existing within the same population, and often this diversity is actively maintained, selected for.
Polymorphism. It sounds pretty complex. So the entry then digs into what it calls classical ecological genetics and this thing called the puzzle of polymorphism. What were these early researchers looking at, and why was it a puzzle in the first place?
Early on, researchers were really into these really obvious outward differences, the kind you could spot easily, like the different colors and patterns on butterfly wings or all the variations you see in snail shells.
Yeah.
Their basic assumption was that these visible differences were caused by variation in their genes. And the puzzle was figuring out how these multiple forms, these polymorphisms, could stick around in a population.
Okay.
It wasn't enough for a new genetic variant to just show up. There had to be a reason why it didn't just vanish or become the only version.
I see.
Like, why does diversity
Or
persist?
right, right.
That was the puzzle.
And so that's where E.B. Ford comes in, right?
Yes?
He's a big name in ecological genetics. What was his take on all this genetic polymorphism stuff?
Ford gave us a really important definition of genetic polymorphism. He said it's the occurrence together in the same area of two or more distinct forms of a species in such proportions that even the rarest of them can't just be explained by random mutations popping up.
Okay, interesting.
So right off the bat, his definition points to a more active mechanism. Yeah. Something more than just random chance is keeping these different forms present at significant frequencies, and that something is likely natural selection.
So he was pretty convinced that these visible variations were practically always genetic, shaped by selection. What made him believe that so strongly?
His reasoning, which was rooted in early population genetics theory, was that in big populations, it's just super unlikely for a new mutation to have absolutely zero effect on fitness.
Okay.
Like it's not going to be completely neutral, neither helping nor hurting the organism. If a mutation wasn't truly neutral, selection would either make it the dominant form over time or wipe it out completely.
Interesting.
So if you see a relatively common, distinct variation hanging around, Ford argued that it was strong evidence that natural selection was actively working to keep it there.
Makes sense.
He even said that even if a trait controlled by a single gene was only found in 1% of the population, it must have been favored by selection at some point in the past.
Wow, that's a pretty bold statement. The entry also talks about transient versus balanced polymorphism. Could you break those down for us? And why was Ford initially skeptical of transient polymorphism?
Sure. So transient polymorphism is what you'd see when a new beneficial mutation is spreading through a population. It gradually replaces the older form. A balanced polymorphism, on the other hand, is when two or more forms stick around for the long haul. And that's because there are opposing selection pressures keeping them in balance. Freud was skeptical of transient polymorphisms early on because he was convinced that natural populations were already perfectly adapted to their environments. All thanks to natural selection working its magic over long periods. The idea that a brand new mutation could suddenly come along and offer a big advantage seemed less likely to him.
I see.
So he leaned towards the idea that the polymorphisms we see in nature were mostly balanced.
Now, the entry mentions sex ratios as an example of balanced polymorphism. Can you explain that a bit more? And why is it called a unique case?
Right, the whole thing about roughly equal numbers of males and females in most sexually reproducing species, that's a classic example often used for balanced polymorphism.
Hmm.
R.A. Fisher famously argued that if one sex becomes rarer, individuals that produce more of the rarer sex will have an advantage because their offspring will have more mating opportunities.
Ah, that makes sense.
Right, and this pushes the sex ratio back to that 1.1 equilibrium.
It balances out.
Exactly. But the entry points out that the forces behind sex ratio determination are kind of specific and work through this pretty simple feedback mechanism.
Okay.
It's different from those other balanced polymorphisms, like the ones with color patterns or enzymes, which are influenced by more complex factors that change across environments and life stages. Ford himself saw the sex ratio as being maintained by a sort of built-in genetic switch, not so much by diverse ecological factors influencing other polymorphisms.
So the big question for these early ecological geneticists was figuring out how natural selection could keep these balanced polymorphisms going. Like how it exerts different effects on fitness depending on the circumstances. Is that right?
Exactly. They were trying to understand how selection could work differently depending on the sex of the organism, what stage of life it was in, where it lived, even what time period it was to maintain those multiple forms at fairly stable proportions within a population.
Now, the entry talks about population size and this debate between selection and random genetic drift. It seems like early ecological geneticists had a different idea about typical population sizes compared to some theoretical geneticists.
Right, that's a major point of historical disagreement. Theoretical geneticists like Fisher, they often built their models assuming huge population sizes.
Okay.
Fisher even said that sometimes you could consider the whole global population of sexually mature individuals as the relevant population size. So in these massive populations, random genetic drift, which is just those chance fluctuations in gene frequencies from one generation to the next, that would be super weak. Natural selection would totally dominate how genes change.
But the ecological geneticists, the ones out in the field observing real populations, they saw things differently, didn't they?
Totally. Field researchers, they were using methods like mark recapture, and they'd see huge swings in population sizes.
Really?
Oh yeah, sometimes like orders of magnitude different.
That's a lot.
They realized that small, isolated populations were pretty normal in nature.
Interesting.
And despite this, Ford and the others in that classical ecological genetics group, they still didn't think drift was that big of a deal. Their logic was that natural selection would be extra strong when populations were declining,
Okay.
basically overriding any drift effects. They thought there was this inverse relationship between how dense a population is and how strong selection becomes.
So when populations shrink and things get tough, selection gets more picky, weeding out the less adapted variations.
Exactly. And when the population is growing, lots of resources, less pressure, selection would chill out a bit, allowing more variation to hang around.
That makes intuitive sense.
Ford also famously claimed that the advantages given by genes in the wild were often pretty big, maybe even over 25%. And if the selection coefficients are that high, then even in moderately small populations, selection would win over drift.
Wow.
He also suggested that the very nature of the selection pressures would shift as population density changed. Makes sense, right? Like the challenges for a rare individual are going to be different from those when its species is everywhere.
Makes sense.
He said, an organism's needs for survival aren't static. They change depending on its population size and what other species are around. This idea of fluctuating selection pressures led him to be pretty critical of Sewell Wright's shifting balance theory of evolution.
Now, Wright's shifting balance theory, that's a complex one. Could you explain in simpler terms why Ford was so against it? And what was his alternative view on how population structure affects evolution?
Wright's theory basically says that in a population that's split into smaller, isolated groups, drift within these groups can lead to new combinations of genes. So one group might stumble upon a really beneficial set of traits. It's like reaching a higher peak on a fitness landscape. Then, if that group gets to that higher peak, migration could spread those good genes, and competition between groups would favor the ones that are better adapted overall.
Okay, I'm following.
Ford thought this whole multi-stage process was a bit too convoluted.
Right.
He preferred emphasizing local adaptation driven by selection within these subdivided populations. He thought that if populations are geographically separated, they can adapt to their specific conditions much faster than if they were all mixed up. However, he did agree that a little migration was necessary to keep things diverse and prevent too much divergence.
Okay, that makes sense.
But not so much migration that it would erase those local adaptations.
I see.
He even hinted at how interactions between genes, epistasis, might help with those local adaptations.
Now, this leads us to the famous Panaxia dominula moth study, which became like the battleground for this whole selection versus drift debate. Can you set the stage for us on this long-term research project?
Right, so this was a huge deal. The long-term study of the scarlet tiger moth, Panaxia dominula, and how its wing colors varied in one specific population.
Okay.
Fisher and Ford did this over many years using mark recapture techniques. Their whole point was to see if the year-to-year changes in the frequencies of these different color forms were mainly due to natural selection working in different ways, or if it was all just random drift in a small population.
So what did they conclude after all that research? They seemed pretty against drift being a major player.
Oh, they were totally adamant.
Wow.
Based on their statistical analysis of how the different moth-colored genes changed over time, they said natural selection was the main force.
Oh.
And they weren't shy about it. They said their findings were fatal to the theory, which ascribes particular evolutionary importance to fluctuations in gene ratios that might happen by chance in small, isolated populations, which was a direct hit at Wright's ideas.
They didn't hold back.
They were convinced that their study was like the first real test of survival and selection in a wild population, and they said it showed zero support for the idea that chance fluctuations were important in this evolutionary process.
That's a pretty strong dismissal. What did Wright say to all of this?
Wright had some strong counterpoints. First, he stressed that his shifting balance theory wasn't just about drift.
Right.
It was about multiple evolutionary forces working together, not just selection or drift alone.
Okay.
He wasn't a fan of that either way of thinking. He also questioned their methods, pointing out that they used older data collected with less rigorous techniques, and he said their gene frequency estimates could be off due to sampling error. But his biggest argument was that explaining the changes they saw, just with selection, would need unrealistically large and fast shifts in the selection coefficients for those moth colors.
Yeah.
Like from being almost deadly one year to giving a big advantage the next. And Fisher and Ford didn't have any ecological evidence to back up those kinds of dramatic shifts.
And Fisher and Ford, they weren't exactly open to those criticisms, were they?
Not really.
Wow.
In their reply, they basically stuck to their guns and even coined the term Sewell-Wright effect as a synonym for random genetic drift.
Oh, wow.
They basically made it sound like drift was just this singular chance-driven thing completely opposite of the power of selection, which wasn't a fair representation of Wright's much more complex view of evolution.
It's kind of funny how those things stick.
I know, right?
Yeah.
Now, Ford revisited the Panaxia data later on. Did his thinking change at all with more time and data?
Ford did look at the Panaxia data again, with more data points. And while he still disagreed with Wright and held on to his selection-heavy view, he did admit that the advantages tied to those rarer genes seemed to fluctuate quite a bit over time. But he still didn't find the connection between selection strength and population size that his own theory predicted. So even with more data, he stayed firmly in the selection camp.
So this Panaxia study, it seems like it didn't really settle the debate, maybe even made it hotter. And the entry mentions some later findings that complicate the initial interpretation of that moth data.
Yeah, now we know a few things that change the story.
Hmm.
Turns out, how those moth wing colors actually show up is sensitive to the temperature when they're larvae.
Interesting.
So some of that variation they saw might have been due to environmental effects, not just genes.
I see.
That means their gene frequency estimates might not be entirely accurate. Plus, newer evidence suggests that the actual breeding population of those moths was probably smaller than they thought, and those selection coefficients might have been lower too. So basically, the original arguments against drift in that study aren't as strong as they initially seemed.
Makes sense. So focusing on those obvious polymorphisms, while important for starting the field, had its limits. This brings us to the molecular revolution and how studying DNA sequences completely changed this whole debate.
Exactly. Studying those obvious polymorphisms meant focusing on traits that were probably already being shaped by selection, simply because you could see them and they varied. But then came molecular genetics, and suddenly you can look at allozymes, which are different protein variants produced by different alleles of a gene.
Okay.
And this gave us hope for a less biased view of all the genetic diversity within populations, including the stuff that didn't have obvious effects on appearance.
And what did those early molecular studies reveal about how much genetic diversity there really is in natural populations?
They found a ton of polymorphism, way more than expected. Across many species, a big chunk of gene loci had multiple forms, and individuals were heterozygous for a lot of their genes. And remember, this was just based on electrophoresis, which only picks up some of the variation.
Right.
The real amount was likely even higher. The big takeaway was that there was way more genetic diversity than we ever imagined from just looking at outward traits.
But this huge amount of diversity created a new problem, the cost of natural selection. Can you explain what that is and how it led to Kimura's neutral theory of molecular evolution?
The cost of natural selection, or Haldane's dilemma, is this idea that if selection is always busy keeping all these polymorphisms balanced, the number of individuals that would have to die off would be crazy high.
Oh, wow.
Too high for most species to handle, given how often they reproduce. The amount of death needed to weed out the less fit variants across so many genes seemed impossible. So Umatu Kimura proposed his neutral theory as an alternative way to explain all this molecular polymorphism.
What was the main idea behind Kimura's theory?
Kimura said that most changes in DNA and protein sequences aren't driven by selection acting on beneficial mutations.
Interesting.
Instead, it's about random genetic drift of mutations that don't really affect the organism's fitness. They're neutral. He argued that even slightly beneficial mutations have a low chance of sticking around, especially if their advantage is small, which is often the case at the molecular level. He said the rate of this neutral molecular evolution should be about the same as the neutral mutation rate.
This must have been a huge challenge to the whole idea that practically everything about an organism is shaped by natural selection.
It was super controversial. At the time, the idea that selection ruled everything was the dominant view in the field. Kimura's theory was like a bomb, suggesting that chance, in the form of drift, was far more important than many biologists wanted to believe.
So then came DNA sequencing. How did that change the debate? Did it give more support to either side?
DNA sequencing let us see variation in incredible detail. We could compare evolutionary rates at different spots in the DNA, like the ones that code for amino acids, replacement sites, versus the ones that don't, silent sites. Neutral theory predicted that silent sites, because changes there don't change the protein, should evolve at a rate close to the neutral mutation rate.
So what did the sequencing data actually show?
Molecular studies consistently showed that silent sites do indeed evolve faster than replacement sites. This backed up the idea of purifying selection, where bad mutations that change the protein are eliminated, keeping the protein sequence conserved. We also found these things called pseudogenes, which are basically dead genes. They're not under pressure to maintain their sequence, so they give us a good baseline for the neutral mutation rate. This further supported the idea that changes in functional genes are limited by selection.
So it's more complicated than just selection or drift, right? The entry also talks about selective sweeps and the reverse footprint of balancing selection. What are those?
Those are specific patterns we see in DNA sequences that can be like fingerprints of selection.
I see.
A selective sweep happens when a super beneficial mutation shows up and quickly spreads. As it does, It wipes out the variation around it, leaving a mark of recent positive selection. Balancing selection, where multiple forms of a gene are kept around, can do the opposite. It can increase diversity around the selected site because those different forms have been there longer, giving more time for neutral mutations to build up. Sometimes this even creates trans-specific polymorphisms, where the same variation is found in different but related species.
So it was preserved over really long timescales.
Exactly.
That's amazing. So while molecular data supported parts of neutral theory, it also showed how selection leaves its mark on the genome in ways you wouldn't see just by looking at an organism.
Exactly. It's not just about the traits you see, it's also about the underlying genetic code.
The entry mentions something called codon bias as evidence of selection acting on seemingly non-functional sites. Can you unpack that for us?
So codon bias is when we see a non-random use of synonymous codons within protein coding genes.
Remind me, what are synonymous codons again?
Oh, right. Synonymous codons are these different three-letter DNA sequences that all code for the same amino acid. So if things were purely neutral, it shouldn't matter which synonymous codon is used. But we see that some codons are consistently preferred over others.
Okay.
And this suggests there might be some subtle selection going on. Maybe it's about the efficiency of transcription or translation. The point is, selection might be operating at these super fine-grained levels within the genome, not just choosing which amino acid gets used, but even affecting the DNA sequence used to code for it.
It's amazing how this whole debate has kept going and has gotten even more sophisticated as we've developed new tools and data. The entry finishes by drawing a parallel between E.B. Ford's early emphasis on selection and a similar statement made by E.V.I. Tarnevo much later during the molecular genetics era.
Yeah, it's pretty striking. Even though the focus has shifted from looking at traits to analyzing DNA sequences, and even with neutral theory becoming a big deal, this core question about selection versus drift is still central to evolutionary biology. Nevo's statement, made decades after Ford's work, basically reiterates that selection is the primary force shaping biodiversity, even though other factors like drift also matter. It highlights how central selection still is to our understanding of life's diversity.
So we've gone from moth colors to entire genomes, and while the molecular revolution brought new ideas and challenged the selection-centric view, selection's influence at all levels is still a major theme in how we understand evolution.
Absolutely. And for you, the listener, the key is understanding just how complex these evolutionary forces are. It's not a simple this or that between selection and drift. It's this dynamic interplay where random events and small populations or seemingly neutral mutations can have lasting effects alongside the powerful force of natural selection, which can shape visible traits and even influence the very code of life itself.
And that brings us to a final thought. Given all this debate, and with all the new insights from molecular data and ecological studies, how might a more nuanced view of selection, drift, and other evolutionary forces change how we understand adaptation and the future of biodiversity in a world that's changing so fast?
It's a huge question that affects how we see and interact with the world around us. We encourage you to keep exploring these concepts. Think about how they might apply to current environmental challenges like conservation and protecting endangered species. Even consider how they relate to human health and disease. Unraveling life's diversity is an ongoing journey, and there's always more to discover.